Thanks again for the presentation. And next we'd like to turn to an update on the oncology toxicology project, and that's Lori.
So I'm Lori Manassian. I'm a medical oncologist. I am the Deputy Director for the Division of Cancer Prevention at the National Cancer Institute of the NIH. I am also under a Memorandum of Understanding working with the Reagan-Udall Foundation specifically on this project, the tyrosine kinase inhibitors and non-QT cardiotoxicity. And unlike IMEDS and the Critical Path to Tuberculosis project, this project really is just getting off the ground. So the problem that we are trying to address is that the treatment with tyrosine kinase inhibitors, primarily for cancer patients, affect the normal function of cardiac tissue and other tissues than just the malignancies. Patients that now have been successfully treated with cancer, either their disease is in remission or they are true survivors, are now living long enough to develop late effects, and one of those late effects is cardiotoxicity. These effects often are not readily apparent. Some of them are immediate, some of them are moderate, but some of them also are late effects that really were not anticipated through the usual toxicology screens that are done under early development. So because in oncology we have a higher tolerance for risk to benefit, where the patients and the community are able to tolerate a little more toxicity than other drug development agents, we have had a long history of capturing adverse effects quite extensively and mitigating the toxicities to the best of our abilities. The potential, when we say cardiotoxicities, it is a combination of very specific cardiac adverse effects to include heart failure, left ventricular dysfunction, conduction abnormalities, some arrhythmias, some QT, acute coronary artery syndrome, myocardial infarction and angina, and then thrombosis and hypertension, which is often seen with the VEGF inhibitors. So cardiotoxicity specifically is a concern, as I had said, because of the issue of our long-term survivors. We're fortunate enough in the oncology community that we have more and more patients surviving their cancers. And while the acute toxicities specific to cardiac can affect the development of these agents in early development and even in late development, often the late stage and the chronic toxicities can affect survival, morbidity, and quality of life. And in particular, as we have a growing cadre of cancer survivors, we have been able to identify some of these later toxicities. And this particular graph was published in Breast Cancer Research in 2011 and demonstrates that for early stage breast cancer patients, if they have survived and not had a recurrence of their breast cancer for at least 8 or 9 years, around the 8 or 9 year time point, now the risk for dying switches from their breast cancer to cardiotoxicity, to cardiac disease. And some of this is because of the accelerated cardiac toxicities that are seen from the very therapies that they're getting. Why focus on the tyrosine kinase inhibitors? Well, there are more than 20 already approved, primarily for the use in cancer treatment. There's a large growing number of these TKIs in under development. In fact, I believe there are some 200 agents that are currently under development. The FDA is in the process of trying to look at mechanism-based drug safety assessments. And since these agents are designed and developed to be mechanistic in their level of activity, in order to best understand their toxicity, we need to rethink some of our toxicity assessments, especially in early development. The TKIs present a really wonderful opportunity for a pilot project to be explored through the Reagan-Udall Foundation by the FDA for a systems biology framework to understanding toxicity. And as you can see from this particular map, there are over 180 current clinical trials worldwide looking at tyrosine kinase inhibitors to date. So why a systems biology framework? or approach. Systems biology in particular is an emerging scientific discipline that blends biology with computational methods and modeling. It involves understanding how the effects are mediated across increasingly complex levels of biologic organization. There's the molecular level, there's the signal transduction, or the mechanisms that your signaling is affecting. There's the specific cells, there's the organ and the tissue level, and then there's the actual person level. In this case, understanding the actions of these drugs, which originate at the cellular level, may actually have crosstalk and have effects that result in symptoms that may not be readily apparent in our early clinical trials. So we need some new tools, some new methods, a theme that echoes across all three of the Reagan-Udall projects currently. And a systems framework would be particularly useful for attacking this particular problem. And in particular, this diagram shows the different hierarchies from the person level, the organ level, the tissue level, the signaling levels, as well as the molecular level. And at the bottom is the levels of hierarchy, so that it's not enough to study just the individual complex levels themselves, but you need to have a framework to link them together and hook them together and better understand how they're interconnected. And that's the ontology, and that work is being done currently through the FDA and several activities. So why Reagan-Udall? Well, this particular project requires truly a multidisciplinary, multi-sector, and multi-stakeholder approach. There is a tremendous need to have not just the government, not just FDA, but NIH and governmental agencies, academia, industry, and patient groups involved. We need expertise from the cardiologist, the oncologist, pharmacology, toxicology, the systems biology, the computational folks, bioinformatics, and mechanistic modelers as well, to come together to even think about the framework for how to put together this project. We need access and the ability to share heterogeneous data. The data that you generate from studying each of those different levels is very different, and how to link them and think of them together and use them across the different levels becomes a very important question. This work would be conducted under one roof through Reagan-Udall. Reagan-Udall would be the facilitator, the umbrella under which to bring people together, to think about these, to bring their expertise together towards a very specific project. The work would be an integrated program, clearly working with FDA employees as well as gathering their expertise. And Reagan-Udall would focus collaborations on concrete outcomes. Ultimately, the development of predictive models would be the goal. Specifically, the ultimate goal would be to be able to have a model or series of models that would be useful in early and middle drug development, so that we could identify different levels of cardiac toxicity and be able, presumably, to mitigate it or to address it. Who would find these findings useful and why? Well, clearly the FDA needs new tools to be able to understand the safety signals. Regulated industry obviously wants to be able to predict toxicities early, both to be able to rule out drugs for early development, but also to consider how to then follow patients in the very earliest studies so that we can understand the effects, so that we can understand what the patients are, understand the baseline patient factors that may accelerate or exacerbate some of these findings as well. And then there are other stakeholders, both research scientists in cardiology, industry, oncology, and the government, who would be wanting to understand these particular pathways better and therefore help identify ways to treat disease and the toxicities. So the TKI, the tyrosine kinase inhibitor cardiotoxicology program is one that is currently being started. The program's key features are to potentially to improve the quality and cardiovascular safety profile of the tyrosine kinase inhibitors, enhancing the mechanistic understanding of these critical agents in the treatment of cancer, and uncovering molecular and network pathways along which these drugs and biologics operate. We don't fully understand. We know that targeting some of these tyrosine kinase pockets are important. We know that, or at least we think they are important in the driving of oncologic growth. We don't always understand what the effects, both short-term and long-term, are. So Reagan-Udall's affiliation with the FDA allows and enables alignment with the FDA needs. There's both a need at FDA for systems pharmacology. There's also expertise there that we would like to tap into, and are actively working with FDA employees in the development of this so that it suits their needs as well. And I'm happy to take any questions.
Thanks, Lori, for that overview of the predictive toxicology program. Any comments or questions? Or Ellen, go ahead.
Yeah, thank you, Lori. This is a project that I'm particularly excited about. It started at our friends Brookings conference, 3-4 years ago? And we worked on it with MD Anderson and others for a while, and it was clear that this really would be a perfect project for Reagan-Udall, because obviously toxicity is an issue in cancer treatment, and we tolerate a lot because patients want to live. On the other hand, we're living longer and longer and longer, and we need to understand what these side effects are. So this is something that I think would be wonderful for us to do, and I really hope that we can get this started. And now that we're kind of transitioned with the OMOP, this would be ideal, and this does require all stakeholders to be at the table. So this is, I just want to stress, particularly important for patients.
Other comments from the board? Any comments or questions from all of you who are here? All right, well, thanks very much for that overview.